Hello, my lovely anatomists and physiologists. Michele here, and we are going to spend a little bit of time being anatomists and then moving into our role as physiologists here as we look at the sensation of hearing. So now you're going to actually begin to believe me when I say a special sense is special because of the complex anatomical structure, right? We didn't really see that so much in olfaction or gustation, but we'll definitely see that here. Now we're looking at, of course, our ear when it comes to hearing, and these are structures that we've looked at as part of our lab work here to begin. And so I have images of our models that we have in the lab. And so let's actually go through and name a few structures, and then we'll try to build in our physiology into that. So what we see in this top corner here is our fleshy part of our ear. This is that external ear piece that's sticking out. It's got a lot of cartilage and skin, and this structure is helping to really funnel sound waves. And so we can call this the auricle is the most accurate name, or we can also refer to it as the pinna, although the pinna is really used for ears that can, for external ears that can really move around like a cat ear. And so the auricle is helping to funnel sound waves into this passageway, which we can see right here. And so we can call this passageway a few different names. I like to say external auditory canal. You might also see it listed as the external acoustic meatus. So there the terms auditory and acoustic are both referring to sound waves, and then the terms canal and meatus are referring to the same thing, that passageway type structure. Now, it is this external auditory canal that's funneling the sound waves. It's aligned with ceruminous glands that produce our cerumen, which is our name for earwax. These two structures, the auricle and the external auditory canal, make up what's called the external ear. And so then that takes us to the middle ear, which we can see here. When we look at the middle ear, we begin with the structure called the tympanic membrane. So we can see it up here. The tympanic membrane is our name for our eardrum, which is also right there. And so you can very much imagine this thin membrane here, like a drum. Okay, so the external auditory canal is a passageway that carries sound waves to this membrane here, the tympanic membrane, and that's the beginning point of what we call the middle ear. And so from the tympanic membrane, we can then see the three what are called auditory ossicles. And so the term ossicle means like a little bone, and we have three. So we see attached to the tympanic membrane, we have the malleus. Right beside the malleus, we have the incus. And then connecting directly to the inner ear, we have what's called the stapes. And so I've drawn mine in there because it looked like it was actually broken. So right here, we're seeing the malleus, and then maybe right there is the incus, and then the stapes. So we have these three small bones. They're connected in exactly that way. And so what we're seeing is that the air waves, the sound waves, I should say, cause the tympanic membrane to vibrate. And the malleus is attached to the tympanic membrane. So if the tympanic membrane vibrates, so does the malleus. The malleus is attached to the incus. So if the malleus vibrates, so does the incus. And then the incus is attached to the stapes. So if the incus vibrates, so does the stapes. So you have these three little tiny bones all in the middle ear that's essentially transferring the vibration from the sound wave ultimately to a pressure wave, to the fluid that's in the inner ear. So that stapes is knocking up against what's described as the oval window, which is taking us into the inner ear. When we're looking at this particular model, we can see in the inner ear, we have the cochlea. The cochlea is kind of this curled, kind of snail shape looking, snail shell shape looking structure. We have this kind of bigger space sort of in the middle of the inner ear called the vestibule. And then we have 3 fluid-filled passageways called semicircular canals. Now we're going to be talking about these inner ear structures, the cochlea, the vestibule, and the semicircular canals are all detecting different sensations. See, because our topic for this particular presentation is hearing, we're going to be focusing in on that structure called the cochlea. In this particular image, we are back to looking at the cochlea. We're looking at that kind of curled shell structure. And what we can see is the very outside of the cochlea is lined with bone, and then you have the inside of the cochlea is lined with a membrane. So we can go back to this picture so I can draw for you. When we look at our inner ear, let's draw a small circle and then leave a space, and then let's draw a bigger circle. And let's label that larger circle the bony labyrinth. Let's label the inner circle the membranous labyrinth. And then what we'll see is in between the bony and the membranous labyrinth, we have a fluid that we call perilymph. So the lymph part of the name just is like a name for liquid or fluid, and peri is around. So the perilymph is the fluid in between the bony and the membranous labyrinth, is around the membranous labyrinth. And then inside the membranous labyrinth, let's label that as endolymph. So the endolymph is the fluid that's going to be on the very inside of the membranous labyrinth. Okay. And then let's go ahead and label that drawing that we did of our large circle and small circle. Let's call all of that our inner ear. And if you want to remind yourself that the inner ear includes the cochlea, the vestibule, and the semicircular canals, you might want to remind yourself of that. Because again, we're focusing on hearing, we're going to be taking a more close-up look at that cochlea, which again is sort of that snail shell-shaped structure of your inner ear that's detecting hearing. And we can see in this close-up view that we have that bone, that bony labyrinth, right? And then we have this inner tissue here with our membranous labyrinth. So what I want to do in this first picture is we're seeing you have a duct above, labeled here as the scala vestibuli. We can also call it, instead of the scala vestibuli, excuse me, we can also call it the vestibular duct. And then we have this opening called the Cochlear Duct, the Cochlear Duct. And so that one is actually going to be full of our endolymph. That's essentially our membranous labyrinth. And then below that, we have the scala tympani, which can also be called the tympanic duct. Now, both the vestibular duct and the tympanic duct are full of perilymph. If you look at this diagram down below, now all of these diagrams are taken from your textbook, your OpenStax textbook, and I've brought 3 different diagrams together onto this one screen. If you look down below, you're seeing your scala vestibuli at the top, you're seeing your cochlear duct in the middle, and you're seeing the scala tympani at the bottom. Remember, this scala vestibuli is the same thing as the vestibular duct. You have your cochlear duct, and then you have your tympanic duct. Now, the vestibular duct and the tympanic duct are named because this is the movement of the sound wave that gets turned into a pressure wave. So what we see happening, remember, is you have a sound wave traveling in through your external auditory canal, causing your tympanic membrane to vibrate. When the tympanic membrane vibrates, so does the malleus. When the malleus vibrates, so does the incus. When the incus vibrates, so does the stapes. And the stapes is actually knocking up against a structure on the cochlea called the oval window. And so what this does is this creates the pressure wave in the perilymph. And so the vestibular duct is named because that movement of that wave is away from the oval window and heading towards the vestibule. But because it's a coiled structure, then the pressure wave comes back toward the tympanic membrane. And when it's coming back towards the tympanic membrane, we call that the tympanic duct. And so just like a tide goes out and then comes back in, just like this pressure wave goes out towards the vestibule, it comes back in towards the tympanic membrane. So you have another structure on the cochlea called the round window. So the oval window is how we get the sound wave translated into a pressure wave, but then you have to have some kind of little release of that, and that's happening at the round window. Now the cochlear duct is really what we can highlight here, because the cochlear duct contains what we call up here the organ of Corti, or you might also have it listed as the spiral organ. So again, that depends on what reference material you're using. And there are really 3 important structures to the organ of Corti, or the spiral organ. This includes our tectoral membrane. So do tectoral, think about this as being on the top. And then do your basilar membrane, maybe think about this as being on the bottom. And embedded in the basilar membrane, you have your hair cell. And your hair cell is your receptor cell. So let's go up to our table that we've been working on. And you can see here that I have been copying and pasting in those afferent pathways. And so it's making my table look a little bit crazy. And so let's go down to where we have hearing. And so for hearing, we're going to be typing in our sense organ. And so here, I think I would be typing in the cochlea, which is that particular part of the inner ear that's involved in hearing. Of course, all the other structures, meaning the auricle, external auditory canal, tympanic membrane, the 3 auditory ossicles, those are also going to be important as well. But it's the cochlea where we're actually going to see our receptor cell located, and we call the receptor cell here a hair cell. So let's go back to our pictures and take a look at that in more detail. And so over here in this blown up picture, we're looking here at our spiral organ, or remember we can call it our organ of Corti. And at the top, remember we have our tectoral membrane. And at the bottom, remember we have our basilar membrane. And here we have our hair cell, and we see that it's embedded in some epithelial cells. Now the hair cell, remember, is our receptor cell, and it's called a hair cell because it has these special cilia extending up from the cell. So remember that when we talk about villi or microvilli, we're talking about just extensions of the cell membrane that increases surface area. When we're talking about cilia, these are modified for movement. Now we actually refer to these as stereocilia. And what we see happening is that the hair cell has these stereocilia. The hair cell is synapsing onto sensory neuron. The hair cell is embedded in the basilar membrane, and its stereocilia are sticking up where that tectoral membrane can make contact. So when nothing's happening, when it's quiet, there's going to be space in between the stereocilia and the tectoral membrane. However, when there's a sound wave that's vibrated the tympanic membrane, that's vibrated the malleus, the incus, the stapes, when the stapes knocks against the oval window and you get that pressure, and the pressure in the perilymph, that pressure wave is going to cause movement through first the vestibular duct and coming back through the tympanic duct, and that's going to cause movement of the basilar membrane. And if the basilar membrane moves, then those hair cells are getting pushed up against that tectoral membrane. And if those stereocilia bend, then we're going to see signal being sent on to the sensory neurons. And these sensory neurons, notice, are coming together to form the cochlear branch of the vestibulo-cochlear nerve, which is cranial nerve number 8. Okay. To finish up olfaction, let's put that together in our pathway. All right, so if I'm tracing out my hearing pathway, I'm going to say sound waves are going to be collected by auricle, funneled into the external auditory canal, or remember, you can call this the external acoustic meatus. This is going to cause vibration in the tympanic membrane, malleus, incus, and stapes. The stapes knocks against oval window of the cochlea. So now we've transferred the sound waves into pressure waves. So now we have pressure waves of the perilymph moving through the, let's say, the vestibular duct. So we're going away from the tympanic membrane towards the vestibule, and then back toward, and then back through the tympanic duct. Okay, so this is going to cause movement of the basilar membrane. That's of the spiral organ or organ of Corti. So now we're going to have hair cells bump against the tectoral membrane. That's also part of that spiral organ. This is going to cause the stereocilia to bend. The hair cell is depolarized. And And that depolarization then is transmitted onto the sensory neurons. And those sensory neurons together make up the vestibular cochlear. Forgot the L after all that. The signal goes to the brainstem, and then to the thalamus, of course, and then to the auditory cortex, which is in the temporal lobe. Now, two things to pick up here. We have what's described as frequency. Frequency is talking about pitch. So how deep it is, or how high pitched it is, how squeaky. So frequency is determined by the location along the basilar membrane. So those pressure waves are moving through the cochlea, and then depending on the pitch, that's where we're going to see the bumping of that basilar membrane. And then if we want to talk about volume, so of course this is how loud it is. This is going to be talking about more like the amount of distortion. So if it's a very loud sound, then it's like a vigorous bumping up against the tectoral membrane. So this is how really loud sounds can be very damaging to your inner ear and your hearing. Okay, that's it for this sense. We're going to move on next to the other parts of the inner ear. So stay tuned. As always, take care of yourselves and each other.